Good day, this is Professor Will, CBMD PhD. Today is Thursday, June 10th, 2021. It is 7:16 and 11 seconds p.m. Eastern Standard Time. For today's research, virus evolution, and feel free to check out my website. So let's hop to it here. A vital tool to study virus evolution. In the test tube. Date June 9, 2021. Source, University of Queensland. Variants of viruses such as that causing COVID-19 could now be quickly studied in the laboratory, even before they emerge in nature and become a major health challenge. Researchers developed a technology to manipulate viruses synthetically, allowing rapid analysis and mapping of new potential virus variants. This technique should give us the ability to answer questions about whether potential virus variants are susceptible to a particular drug or vaccine, even before they emerge in nature. Up until now, we've mostly just waited and reacted to viral variants as they emerge. And in the case of SARS-CoV-2, the world has been hit by Indian, UK, and South African variants. Just to name a few, WHO has now reclassified these variants as Alpha UK, Beta South African, and Delta Indian. Now we can mimic the massive experiment going on in nature, where these mutations pop up due to natural selection. But we can do it safely, in a strictly controlled and highly regulated biosecurity laboratory environment. The UQ developed process uses copies of fragments from the viral genetic material to assemble the functional viral genome in a test tube. This allows scientists to rapidly generate virus variants and assess their potential to evade antiviral treatments and vaccine-induced immunity. Researchers evaluated infection and disease caused by the test tube, made virus and preclinical models to ensure the technology was able to generate authentic viruses. We can now monitor changes in viruses like SARS-CoV-2 and can see which variants may not respond to certain vaccines and antiviral treatments. Researchers can also investigate whether potential variants are more or less virulent in mice and find out which drugs and vaccines will be effective. It is great to finally have this vital tool and start tackling these challenging questions. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and pathophysiology. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.